Thanks, David et al. For organizing the workshop. I'm happy to talk about the work we have been doing in our lab at NIH on exploring microstructure and transport processes in the brain. To give an overview, after working with David's group for a while now, we believe that high field, high slew rate, high gradient MRI scanners really enable like novel microstructure imaging methods from excised tissues to in vivo. I'll describe some examples of quantitative MRI methods that reveal brain microstructure and organization and architectural features. I will illustrate how these measurements of key transport parameters can help characterize the transport processes in vivo. I will also show maps of intrinsic transport properties that can be used as novel imaging biomarkers to follow development, disease, degeneration, aging, and trauma. The holy grail of microstructure imaging is an example is the electron microscopy image of a piece of cerebral cortex of a human provided to us generously by Jeff Lichtman's group at Harvard University. This took several days to acquire, and this is nanoscopic resolution. Our goal is to actually see if we can get this kind of information in vivo in a 40-minute scan. So when Jeff analyzed these EM images of cortex, a lot of things popped out. Different layers of the cortex have different cell sizes. They have different orientation distributions, and they have different shape distributions. And diffusion MRI is kind of uniquely positioned to kind of use the diffusion of water to probe these microstructural properties. It started, in the 90s, Peter, invented DTI, one of the first methods, one of the early methods to look at tissue anisotropy. And you can start looking at the orientation of different fibers in these color maps, and you can start doing some tractography by linking these ellipsoids to really get the wiring diagram of the brain. But DTI has its own limitations that it cannot really resolve crossing fibers, and it only assumes a single compartment. And we have come a long way from DTI. So one of the things we have developed is called map MRI, which basically reconstructs the diffusion propagator, or what Einstein found, that the displacement distribution function in 3D in vivo using very high B values. And this technology subsumes DTI, so you can get all the DTI stains added. We can also get new stains like the zero displacement probabilities and the non-Gaussianity of diffusion, and improved definition for the FA, or the fractional anisotropy. So you could see, comparing to the DTI maps, these maps are show the tissue microstructure in exquisite detail. We applied this method on non-human primates. This is comparing the color maps we got from map MRI, and this is the magnetization transfer ratio, and this is the T1 images. You could see that map MRI really shines compared to these other structural imaging methodology. And at this, we did this scan at 200 microns resolution. And at this resolution, map MRI, you can really start drawing domains of the tissue and compare them with histology. So we can start doing what people would call virtual histology with MRI. And looking at the cortical areas, so you can at these high resolutions, the map MRI starts picking out what looks like cortical layers. Remember I said that these layers have different geometrical, the cells in these layers have different geometrical properties, and the map MRI seems to be catching that a little bit. So you can, the DTI maps look a little bit uniform, but when you start looking at this higher order diffusion processes, you could start picking out bands of tissue that look like layers that seems to align with histology. And we can also start looking at cortical areas, like where different layers transition, which is not visible in DTI, but you can start picking it up in map MRI. And we can also start doing subject-specific cortical parcellation. So you can segment the cortical regions into using these map MRI stains and passing it through an unsupervised learning, like K-means clustering, and you can start getting a subject-specific cortical parcellations. And we also did this on a human excised brainstem, and you could clearly see the map MRI stains, or it really shows like various deep nuclei, which are usually not visible in conventional scans. And next, we cannot get 100 or 200 micron resolution, but we worked with David's group to do this in vivo at 900 microns. So we are kind of getting there, but and you can see that at this resolution, the map MRI and DTI maps show various tracks, and the maps are much richer than what you would get from a conventional 2 mm scan. And we can also start segmenting different tissue types. And if you start zooming in into some of those cortical regions, there are like stripes, that what appears like layers, in these maps. But obviously, we don't have the resolution to resolve individual layers, but there are some indication of it that may need some further evaluation. And with the high resolution, we can easily resolve different crossing fibers and produce tractograms with exquisite detail, and really show these various crossing fibers with these high B values and high resolution that are really made possible by David's scanner. Even though DTI and map MRI are really excellent techniques, they have their limitations too, in that they're not very eloquent in describing heterogeneity. So you can imagine like two voxels having very different microstructural motifs. So in the one is free water, all the ellipsoids are spherical, and the second one could be an emulsion of spheres, like replicating cells. If you look at the diffusion tensor, they actually look the same. You cannot really distinguish them. And for the anisotropic case, if you have two crossing fibers or one fiber having different sizes, they both have the same DTI tensors. So in some sense, there's some ambiguity in here. So we have been working on this technique called DTD MRI, which basically says that, well, let's assume each voxel is composed of a distribution of tensors. And when we start having that kind of description, you can look at the variability of these tensors. So if you look at the free water case, there's no variability. So you can start teasing out these different motifs with the DTD MRI. So for the emulsion type, there's an isotropic variance in the size, so you can nicely capture by this covariance cliff. And you can see the same thing for the anisotropic case. The covariance clearly of the diffusion tensor clearly captures the two fiber populations, and the same for the size distribution. Once we have the diffusion tensor distribution, we can start extracting the size, shape, and orientation heterogeneity measures. So, for example, you can compute the DTI or ODF, which basically shows a single peanut. But then, if you start using this DTD information, you can really come up with what we call as micro ODF, where it shows the two dominant fiber population. So you can also start looking at the distribution of sizes by just looking at the histogram of the mean diffusivity. So this gives additional information that usually you cannot get from DTI. And we actually scanned a macaque cortical tissue at 100 microns, and this clearly shows that if you look at the mean diffusivity map, it's quite uniform because it's well known that the mean diffusivity of tissues are quite uniform in the brain. But then if you start looking at the moments of the size distribution, like the standard deviation of mean diffusivity or the skewness, there are these bands that pop up that really aligns with the histology images we have. I think with DTD MRI, we are really at these high resolutions, we are really able to image these cortical layers. And we actually tried this on Connectome 1 scanner at MGH at 1.2 millimeter resolution. And we also saw some notions of bands, but really the resolution is not high enough for us to really look into cortical layers. But I think we are kind of getting there. So we have been developing different, flavors of DTD, and one of them has been developed by Alexander Avram in our group, specifically tailored for cerebral cortex. So we know that in cerebral cortex, the fibers are aligned primarily in one direction. So if you know the direction, you could just reduce the complexity of the DTD problem into just evaluating the density of the eigenvalues of the tensor. We can basically remove 3 dimensions. And when we do that, we start seeing distributions of mean diffusivity and micro FA, and we start seeing different water pools that are present in the cortical regions. And this is called the cortex MRI. So far, we have been mainly looking at diffusion, but I think MRI is a very rich technology that we can start looking at combining different MRI contrasts by this multidimensional MRI. So we started work combining diffusion with relaxation and to really observe the dynamics of different water pools in the tissue, which are often ignored or invisible in most of the optical microscopy or X-rays and EM techniques. And these naturally require more data to come up with these multidimensional distributions. So I think, the faster, stronger gradients and the high throughput afforded by these scanners really enable these methods to go to the clinic. As an example, we did some work on TBI. So by Dan Benjamini, who was in our group, and now he's a principal investigator at the National Institute of Aging. In a healthy tissue, in a healthy neuroaxon, all the biopolymers are quite confined or compartmentalized in the myelin. But then if you have a disease, if you have an impact TBI, you start these proteins starting leaching out into extracellular space, and there they could interact with the water molecules and really reduce or increase the relaxation times. So we collaborated with Dan Pearl at the USU in Bethesda, and we did some studies on TBI brain samples. And when we did DTI and T1 and T2, these maps are quite uniform. It doesn't show any indication of injury. But then when we start combining, T1, T2, and diffusion, we can really identify injury areas. Like there are some distinct signatures in the multidimensional space we could just map, and that seems to align with this histology image we have for the amyloid precursor protein. So we are really making, with these high dimension methods, making invisible things visible. And we have also expanded that to in vivo, but obviously the scans resolution is very coarse. This is a work by Alexander Avram looking at T1 and diffusion. You could clearly see the different water pools, how they separate in the 2D distribution. Recently, we have been looking at different, transport processes. One of them is the magnetic resonance elastography, which is a technique used to map the stiffness of tissue. So, palpation is as old as medicine for a reason, because, when T1 relaxation, bulk modulus, X-ray coefficients only change by a factor of 2 or 3 in disease, the shear modulus probed by the elastography could vary by orders of magnitude. So it really is a very sensitive method to detect disease or look at function. And neurons, when it comes to the brain, neurons are very sensitive to the stiffness of the substrates they live in. So this is an example of a neuron that is grown on a softer substrate. You could see the extensive branching, and the astrocytes are more smaller. But then when the substrates get stiffer, the branching reduces in the neurons and the astrocytes become reactive. So this could be a very important biomarker for several diseases. The way it works is like there's an external tamper that to palpate the sample that sends in mechanical waves into the sample, and these are the displacement induced by these waves are concurrently measured with the phase contrast MRI, and the displacements are inverted to obtain stiffness. Um, Obviously, the use of a tamper is really challenging because it's expensive, and it requires, yeah, technique to really operate the tamper. It may obviate, it may be contraindicated for certain patient cohorts, like one with TBI or fetuses or infants. So we have been working on this technology called tamperless brain MRE or intrinsic MRE, because with every heart, with every minute, the heart pumps roughly 3/4 of a liter to the brain, and that causes the brain to move and deform. And if we can actually capture that motion, the idea is to use that motion to back out stiffness. So you could, basically take a cardiac cycle and divide them into 10 different bins, and we can do a simple test called standard diffusion acquisition, and kind of retrospectively bin them into each of the individual bins. And when you look at the phase of the MRI, we can get the displacement in 3D. And when you look at the magnitude, we can actually get the diffusion tensor. So we can get both of them at the same time. And once we have the displacement vector field, we could use the momentum transport equations to estimate the stiffness properties. And we had the opportunity to try this on David scanner. These are 1.6 millimeter isotropic scans, and these are the different displacement vectors we measured in the brain in 3D, and these are the concurrent diffusion tensor maps. So the brain is never, throughout the cardiac cycle, you could see the brain is never still; it's always bouncing. And that actually affects the DTI measures too, because different tissues may come in different parts of the cardiac cycle, and that could be a local partial voluming artifact. And since we have the displacement and the diffusion tensor maps concurrently, we can use the displacement to deform the DTI maps and really get like a dynamic deformation MRI. Obviously, these displacements are amplified 40 times to kind of accentuate the motion. But you could see that the brain is really pulsing, especially the wave comes from the heart, and then the brain just expands and contracts, and almost the whole brain deforms. And you could use this deformation to extract the stiffness properties, because a stiffer tissue would deform less compared to a softer tissue. And we obtained these maps, so you can get the main diffusivity from DTI, and this is the shear modulus maps and the bulk modulus maps. So you can look at the mean diffusivity, like in the exercise tissue, is more or less uniform, but then the shear modulus maps really is very heterogeneous. So it starts identifying boundaries, and it's much more richer than the trace. And since we have DTI, we can do tractography and we can overlay these properties. So we can start looking at mechanical properties of fiber tracts with this methodology. And for all these to work, we need good phantoms to calibrate our gradients. And Ferenc Horke in our lab, who recently passed away, was instrumental in coming up with the PVP phantom, which is a shelf-stable polymer, non-toxic, which we use, which our lab pioneered and had been used in several facilities. To summarize, high field, high gradient, fast loaded MRI scanners enable microstructure imaging methods. And we believe that the in vivo mesoscale MRI data can help bridge the top down and the bottom up divide eventually. And measuring key transport properties helps us construct models to understand transport processes and key aspects of physiological function. And these properties could also serve as imaging biomarkers, which may be important in clinical and biological applications. Obviously, this cannot be done by a single person. So we want to thank our wonderful group we have at NIH and our collaborators outside NIH. Thank you very much.